Hello, everyone. This summer, I have been conducting my research in the entomology department in Quinn McFrederick's Bee Lab. My presentation is on the two stressors, Crithidia bombi and cadmium, and their influence on mortality and gut health of the bumblebee, bombus, and patients. I'm sure many of you, if not all of you, have heard at some point that bees are in decline around the globe. But how many of you know why bees are declining? Bees are important insect pollinators that contribute to the biodiversity of our landscapes and agricultural production. Therefore, their decline threatens not only the food that we eat, but the natural world as a whole. The decline of bumblebees is caused by many stressors. Some of those are toxicants, pollution, habitat destruction, and more. Bees don't come into contact with just one stressor throughout their lifetime. Rather, they come into contact with multiple stressors simultaneously. Two very common stressors that bumblebees in particular have to face is the heavy metal cadmium and the parasite Crithidia bombi. Cadmium is a toxicant not only found in the United States, but all over the world. This heat map illustrates how prevalent and widespread cadmium is in the United States alone, with the warmer and red colors illustrating a higher concentration of cadmium in the soil. Cadmium has a wide range of effects on bumblebees, such as increased mortality and reduced fitness and an alteration to their gut microbiota. Crithidia bombi is a parasite that spreads rapidly amongst social bees and primarily affects the gut of their host. Crithidia bombi usually works in conjunction with other stressors to bring out harsh effects on their host. Some of those effects are increased mortality and reduced fitness of worker and queen bees. To try and simulate a real-life situation of bees coming into contact with multiple stressors throughout their lifetime, my specific research is focusing on the two stressors, cadmium and Crithidia bombi, and their synergistic effects on the bumblebee, bombus, and patients. First, we want to discover how the two stressors, Crithidia bombi and cadmium, affect the overall health of the bumblebee. As mentioned previously, Crithidia bombi has been known to reduce fitness of worker and queen bees and work in conjunction with other stressors to have synergistic effects. Cadmium has been known to increase mortality rates in bumblebees all on its own. Therefore, I hypothesize that the two combined stressors of cadmium and Crithidia to have synergistic mortality effects on the bumblebee, bombus, and patients. Second, we investigate how the core gut microbiome of the bumblebee, bombus, and patients influences Crithidia abundance. The microbe of the bumblebee, bombus, and patients has been shown to decrease proliferation of Crithidia infections. Therefore, the microbe-depleted treatment group will have a higher abundance of Crithidia bombi because of the lack of protecting microbes. Expanding on this notion and relating it to cadmium, cadmium has been known to cause dysbiosis to the beneficial gut microbiome of bumblebees. Dysbiosis is an imbalance of an organism's natural microbiome and is usually associated with a range of health effects on that organism. Therefore, I hypothesize that the cadmium-treated bees will also have a higher abundance of Crithidia bombi due to an alteration to their beneficial gut microbiome that protects against Crithidia infections. We started off this experiment with bee maintenance. We received 12 commercial bombus and patients colonies from Copert. Each colony had around 50 or so bees, including one queen. We checked to see how they're doing and if the queen is alive and well. Then we take out the Copert provided pollen log and sugar solution with our own 60% sterile sucrose solution and honeybee pollen patty. We allow the bees to feed freely on the sucrose solution and we change it out about once a week. We provide a pollen patty every other day. We had to work with the bees in a temperature controlled room in the dark and under a red light. We work with them in this fashion because bees cannot detect red light, so they will be less likely to fly and sting us, so it's safer to work with them in this condition. After bee maintenance, we focus on pupae collection. We monitor each colony for cocoons taking them out and putting them into sterile containers. Once the cocoons have darkened enough, we carefully and aseptically pull out pupae from their cocoons and place them into sterile tissue plates. We do this to try and simulate a germ-free bumblebee. As bees metamorphose from larvae to adult, they break down their larval gut and rebuild it. As they emerge from their cocoon, they shed their larval gut, giving us a sterile adult bee to work with. After placing them in the sterile tissue plates and waiting for them to be fully emerged, as seen here, we randomly assign them into one of two treatment conditions, either microbe-inoculated or microbe-depleted. After assigning the bees into microbe-inoculated or microbe-depleted, each group was further split into 4 more treatment conditions. Those treatment conditions are a no Crithidia and a no cadmium control group, an only Crithidia and no cadmium, an only cadmium and no Crithidia, and then a cadmium and Crithidia group to give us a total of 8 treatment experiments. We need about 400 bees to complete this entire experiment. Here's a flow of the treatment conditions. After the bees have fully emerged, we split them into microbe-inoculated or microbe-depleted. The mature bumblebees, worker bumblebees in our colonies, harbor the vast majority of their microbes in their hind gut. Right here. The microbe-inoculated bees were then fed the guts of their nestmates, mixed in with a sterile sucrose and pollen solution. The microbe-depleted bees were just fed a sterile sucrose and pollen solution. From there, we moved on to the Crithidia and sham Crithidia inoculation treatment groups. The Crithidia treatment group received a field realistic amount of Crithidia inserted in a droplet form mixed with sucralose and media. The sham Crithidia inoculation treatment group received just sucralose and media in a droplet form. From there, we moved on to the cadmium and no cadmium treatment conditions. The cadmium group received field realistic amounts of cadmium inserted in their sugar feeders mixed with sterile sucrose. The non-cadmium treatment group just had sterile sucrose inserted in their sugar feeders. The bees were allowed to feed on their cadmium and no cadmium respective treatments freely for 7 whole days. We monitored for mortality every day, and once the 7 days were up, we flash froze the bees in an ethanol and dry ice solution and then placed them in a negative 80 degrees Celsius freezer. As of right now, we're still collecting data on our mortality experiment and have not yet started on the methodology for the Crithidia abundance experiment due to some unforeseen events with our bumblebee colonies. Therefore, I have 2 main predictions for our experiment. The first prediction I have is that we will observe a high mortality rate, which could be due to one of 2 reasons. It could be due to the cadmium treatment alone, because cadmium has been known to increase mortality rates all by itself, or it could be due to a synergistic combination of cadmium and Crithidia, because Crithidia has not been known to increase mortality all by itself, but has been known to have detrimental effects such as mortality when in conjunction with other stressors. My second prediction is that for the Crithidia abundance experiment, we will see a high abundance of Crithidia in both the microbe-depleted treatment and the cadmium-treated bees. This is because the microbe-depleted treatment will not have the necessary microbes to protect against Crithidia proliferation, giving it free range to grow in their guts. The cadmium-treated bees will also have a high abundance of Crithidia because of the alteration to their beneficial gut microbes, which also protects against Crithidia. In conclusion, we know that bees are important. As of right now, it is very common for researchers to focus on one specific stressor and how that stressor affects bee health. But bees do not encounter just one stressor in their lifetime. Rather, they come into contact with many different stressors all at one time. Therefore, it's important for us as researchers to view in a lab-based setting how multiple stressors interact with one another to affect bee health. This experiment focused on the 2 common stressors, cadmium and Crithidia bombi, and how they affect the overall health and gut of the bumblebee bombus impatiens. But there are many other synergistic effects and stressors out there and other bees out there that can be viewed in conjunction with one another to see their effects. This could help us as researchers fully understand how and why bees are in decline. In the future, we hope to finish the Crithidia abundance methodology with qPCR. And then we also hope to see how the heavy metal cadmium and Crithidia bombi affect the core gut microbiome of Bombus patients via Illumina next generation sequencing. I would like to thank Dr. Jessica Purcell and her Six Legs Summer Bridge to Research program and her funding by the USDA NEFA grant, because this has been an amazing experience, and I can't imagine my summer without the RISE program now. I would also like to thank Nee Tran, the RISE coordinator, my PI, Quinn McFrederick, my graduate student mentor, Laura Ledger, and all of my lab mates for welcoming me and helping me in this experience.